this slide summarizes what we've done, I guess, in the last over the last few years. We've translated mesenchymal lineage technologies for a large number of indications, and I'll touch on those in a couple of minutes. We've got a very large intellectual property portfolio of more than 60 patent families, which we believe provides long-term commercial protection for mesenchymal lineage cells very broadly. Our portfolio of allogeneic regenerative medicine product candidates are quite advanced, with at least 5 products now in phase 3 trials or that are phase 3 ready. We've got a very well-characterized understanding of the mechanism of action that supports our clinical translation, and I don't want to underestimate how important this is. Very thorough, meticulous preclinical, in vitro, in vivo, small, large animal models before we ever begin clinical trials and validation of mechanisms. We've established strategic partnerships to support development and commercialization of our product candidates. And again, strategic partnerships are central to the ability of cell therapy companies to get products onto the market. I think for anybody to think that we can do these things on our own and be vertically integrated is nonsense. And so not only do we need partners, but we need strong capital market support and a strong balance sheet. And I think it's important to have, if you can have a couple of $100 million in the bank, you've got a chance of being a pharmaceutical biotech developer. This slide speaks to our competitive strengths as a company in this space. We believe we've got a disruptive technology platform. We've got a broad portfolio of distinct late-stage product candidates. And again, as you heard the previous speaker, process is critical to product candidates. Manufacturing drives, process drives delineation of products. We've got scalable manufacturing capabilities. We use 3D bioreactor processes, serum-free media. That's where this whole field is going if you're going to have products that are able to be scalable in industrial fashion. Strong intellectual property leadership. We've got strategic alliances and a very strong management team, which very recently has been expanded to include people with established experience in pharmaceutical drug development from parallel sectors. Now, our disruptive technology, as we call it, is built around mesenchymal lineage cells, particularly very highly purified, immunoselected earliest progenitors of the mesenchymal lineage, defined on the basis of both surface characteristics, STRO1, STRO3 expression, CD146 is another typical marker, but also genotypic expression. And we believe that you've got to define your cells very well at the very earliest stage. A homogeneous population allows you to control manufacturing, allows you to define mechanisms of action, allows you to understand what the cells do and how to control them and how to potentially change their formulation characteristics in culture. Our intellectual property covers diverse source of cells from the bone marrow, from adipose tissue, and from dental pulp. And we've focused on product life cycle extension so that we're developing next-generation products to follow in the particular areas of clinical development that we're interested in. Mesenchymal lineage cells are present around blood vessels in all tissues in the body, in the adult, where they respond to signals associated with tissue damage. This response includes the secretion of a diverse variety of biomolecules that affect reparative and immunomodulatory mechanisms responsible for maintaining tissue health. So if you understand what these cells do in vivo, you understand where they are, you can understand how they respond to damage signals, and you can start to understand the potential disease states that we're trying to address. In addition to where they're located and what their characteristics are, in addition to what they secrete in terms of tissue repair and neovascularization and etc., they have 2 other very important distinct characteristics that, when combined with manufacturing processes, enable us to develop an allogeneic or otherwise an off-the-shelf use of our product candidates. And these 2 very important characteristics are the fact that these mesenchymal lineage cells can be culture expanded very easily, well, not easily in complex ways, but they can be expanded in very large numbers. And secondly, they appear to evade the immune system through mechanisms of immunomodulation. They don't activate the immune system. Those 2 characteristics means that you can scale up a manufacturing process with from one donor to thousands of unrelated recipients, therapeutic doses that otherwise meet potency assays, release criteria, and batch-to-batch consistency, very much like typical small molecules that the pharmaceutical industry is built on. This is our pipeline chart of products and their stage of development. And what you start to see here, we've put a lot of resources behind development of our product candidates. So first of all, what you see in the left-hand side is what we call our product candidates. We've defined each product by manufacturing process, mechanism of action, formulation, etc. We then have programs. So for each key product, there's a lead program. Some of these programs are in phase 2 or phase 3. But this delineation of products and stratification of lead indications has allowed us to actually prioritize our products and their programs into tier 1, tier 2, and below. And what you're starting to see is an evolution from a startup company with a broad scattergun approach to a much more mature stage biopharmaceutical company that focuses our resources on late-stage product development. We can't do everything. We have to focus on areas that we believe are the most valuable for us and where our technology is the most advanced and has the greatest probability of success. So you see our tier 1 products. There are 3 of them. One that has two nomenclatures, but there are 3 tier 1 products. The MPC-06ID is our lead candidate that's about to commence phase 3 for chronic low back pain in patients with intervertebral disc disease. We've completed a 100-patient phase 2 trial, demonstrated significant benefit in terms of both reduction in pain and improvement in function relative to placebo over a sustained period of 12 months. And if we meet that kind of primary endpoint, composite endpoint of pain and function, the FDA would approve us for a product for wide use for chronic discogenic low back pain. Our second tier 1 product is the MPC-150IM product, which is a product being delivered by catheter into the left ventricle in patients with advanced congestive heart failure. This product is actively in phase 3. It's recruiting across more than 30 sites in the United States right now. It is a 1,700 patient trial it's an adaptive phase 3 design with a couple of interim analyses along the way for both safety, efficacy, and potential resizing. What we're very excited about this program is that the phase 2 trial has demonstrated that a single injection of these cells into the left ventricle of patients with class 2, 3 heart failure with low ejection fraction has resulted in complete prevention of any heart failure hospitalization or death over a 3-year period of follow-up. In comparison, controls who received the sham procedure have had a 30% event rate. Now, that's the primary endpoint that if we're successful in this phase 3, will get us approval from the FDA. So, again, a very substantial body of work has resulted in us being able to move this program into phase 3. It's a very large unmet need that remains unmet despite recent developments in the pharmaceutical industry, such as Novartis's new drug for heart failure. The third tier 1 product is our mesenchymal stem cell product delivered intravenously for children and adults with graft versus host disease. In Japan, that product is partnered with JCR Pharmaceuticals. They filed for regulatory approval in Japan about 6 weeks ago, and we expect that they will get approval sometime during the course of 2015, which would be the very first allogeneic culture-expanded stem cell product approved in a first world regulatory authority, at which point we would be a revenue-generating company. In parallel, the same product is being developed in the US by us. We've had discussions with the FDA and are about to begin the final phase for this product to be launched in the US. We have to do a 60-patient open-label phase 3 trial in children with graft versus host disease. And this is on the back of more than 200 children that have been treated under expanded access scheme in the United States, where a multi-dose injection of this product has demonstrated a significant responder rate and that correlates with a significant improvement in day 100 survival. If we see the same kind of data in the 60-patient trial, we will be able to file for BLA approval in the United States and have the first, again, allogeneic culture-expanded stem cell product approved in the United States. Our Tier 2 pipeline, the most important there, I think, is the product that we call MPC300 IV, and that product is our immunoselected, relatively homogeneous population of mesenchymal precursor cells that is being used intravenously to address diseases of immune dysfunction. The mechanism of action is quite distinct. It's response to pro-inflammatory cytokines, release of factors that then converts inflammatory monocytes and inflammatory T cells to anti-inflammatory subsets. And the two major areas of focus are diabetic kidney disease and biologic refractory rheumatoid arthritis. And we expect to have data, top-line data from these two phase 2 trials in Q1 and Q3 next year, respectively. But I think what you're seeing from this is that this is now a very advanced pipeline that we have prioritized based on real outcomes and real value chain. This slide speaks to the manufacturing facility for our cells. We have a strategic partnership with Lonza, the world's leading biologics manufacturer. We have exclusive access to their Singapore facility for allogeneic cell therapy use. It's a very large facility. It's modular. It can be expanded as our needs expand. And what we're doing there is now commercial scale manufacturing using 3D bioreactors, serum-free media, really in preparation for commercial launch of our products, predominantly in the US, but potentially also in other jurisdictions. And this slide is a very busy slide, but I think it speaks to our broad patent portfolio that's structured along compositions of matter and manufacturing, very important patents around manufacturing, specific therapeutic applications. So for every indication, we have a portfolio, and ultimately complementary technologies and additional indications. And finally, I think, again, this slide touches on the various strategic partnerships that we have in place. Can't do this by yourself. As a company, we've focused on manufacturing as a partnership with Lonza, development path relationships with the NIH and with Quintiles, and at the commercial stage, our partner in cardiovascular disease is Teva Pharmaceuticals. For GVHD in Japan, it's JCR, and we're actively in discussions with other partners in other indications. And finally, this is the slide on news flow for the next 12 months. Very busy, but I think suffice to say that we're focusing on a number of programs. This really speaks to the Tier 1 and Tier 2 programs and their news flow potential over the next 12 months. And I think on that note, I might leave it. Thank you very much.